And welcome to the Antibody Engineering and Therapeutics Digital Week, brought to you by the producers of the online and face-to-face antibody events. My name is Brianna Costafret, and I'll be your host for today's session titled Development of a NK Cell Engager Utilizing Antibodies Targeting a Single Amino Acid Variation. First, I'll cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. If you are experiencing other issues, hit the question mark button to receive assistance. At any time during the presentation, submit your questions into the Q&A window on the left-hand side of your screen. In 24 hours, you receive a link to watch the recording of the session, and also please take a peek at the resource list box on the right-hand side of your screen. Let's now begin by introducing our speakers for today's session. We have Dr. Jordon Wang, COO and SVP of Technology Development for AvantGen, and Indrani Chakraborty, Manager of Applications for Gator Bio. Welcome, Jordon and Indrani. I'll now hand it over to Indrani to begin.
Hello everyone, my name is Indrani Chakraborty and I am the Manager of Application at Gator Bio. Thank you all for joining for our webinar today. And today my topic is accelerated discovery of multispecific antibodies using Gator Bio's BLI technology. For today, I will be introducing you to our company Gator Bio, giving brief information regarding what is the biophysical principle behind our bioanalytical instrument, and also trying to showcase some examples of multispecific antibody characterizations that are done using our different kinds of biosensors and also using our high throughput instrumentations. Now, our company Gator Bio has been in existence for last almost 7+ years, and we are headquartered in Palo Alto, California. And the company has been founded by Dr. Hong Tang and Robert Zook. Both of them are actually the innovator of the BLI technology. And we are, of course, based out of lot of 60+ patent that protects our intellectual properties, as well as we are constantly innovating each and every single day in order to help our current users keeping up with the drug discovery trends. We are, based out in Palo Alto, California, but our presence are everywhere around the globe, where we have offices in Shanghai as well as Basel, Switzerland, and Singapore. Currently, we have 300+ users, and these users are using Gator Bio, different kinds of systems and biosensors each and every day, doing different applications for their analytical development work. Now, what is biolayer interferometry? I know the audience here today who is listening to the webinar are all of you almost are very familiar with the BLI technology, and I'm sure you must be using, most of you must be using one of the instruments in your lab almost on a daily basis. But for very few who are getting to know this biophysical principle today, it's basically based on a very simple everyday phenomenon that we observe, that when light is incidented from one medium to another, there is a change in refractive index, and that's what we utilize in the tip of our biosensor, where everything goes on, all the reactions. So if anything that binds to the biosensors, there will be changes in that refractive index pattern, and that is what is plotted in the y-axis as a nanometer shift against time. So when the advantage of our machine or the BLI principle is that you can see real time binding event happening, and you don't need to put any kind of labels in order to see this interaction happening. We have different biosensors or chemically modified surfaces that helps you see these interactions in real time. Now, how do you do the experiment? So of course, it's not only the biosensors that you can just buy and see the binding event happening in real time, but in order to see or set up your assay, that by working with our instrument, it comes as a package. We have the instrument, we have the several kind of biosensors and consumables. Along with that, the third component, of course, is very important, which is our software. And you have analysis software and the assay setup software. So these three features are that combines provides you an exceptionally very versatile tool for your day-to-day research in your lab. And we are not the only BLI company in the market, but what we did to make even BLI greater is, of course, our proprietary surface chemistry that makes our biosensors as well as our instruments high sensitive compared to our competitor. And this high sensitivity is the fact that we are constantly innovating on our surfaces and also inside our instruments in order to design better optical layers for enhanced sensitivity so that we can expand the dynamic range of the BLI and take it to the next level. So for us, we are compatible because of this new optical layers that we have introduced, the new kind of surface chemistry that we have developed. So we are compatible with a wide dynamic range of molecules, starting from very small molecules to large molecules, thereby helping our current users in their innovation at every stage of the drug discovery process. And of course, the other thing that sets us apart is also our easy and more user-friendly software, which allows you to run your experiment without having to have a very steep learning curve. And along with that, we have also, like currently we provide a wide ranges of biosensors and assay kits in order to facilitate your drug discovery process so that you have so many choices in front of you to try and test and optimize your assay conditions depending on what you are interested. Now, how, or if you have not used a BLI instrument, so how can you think yourself using it? If you think about it, that we are catering to all sorts of users, starting from early discovery research projects to all the way manufacturing and QC. So we have users who are CROs, CDMOs who are doing phage discoveries, hybridoma panning, and all sorts of antibody discoveries that they use our instruments on a day-to-day basis, doing antibody titer, yes or no binding, isotyping, off-rate ranking. They also use it sometimes when you have moved on from selecting those leads to the next step. You can do a bit of binning, you can study your affinity maturation, honing in your lead optimization, as well as you can choose or screen different kinds of buffer when you are doing your developability experiments. Again, when you are into manufacturing QC, we see that our users are frequently using our machines in order to do a batch to batch study in terms of quantitation, glycosylation pattern for their antibodies, etc., just to name a few. So you can see that how versatile the instrument is in a lab, that it can aid your discovery from early to late stage in all to support your research work. Now, today's topic chose multi-specific antibody discovery and development using Gator because when we look at the FDA approvals recently, as well as we see our user base, what are they using it, we realize that slowly the traditional antibody discovery is taking a different shape. So now the discovery has been more focused on discovering small fragments which are part of like an antibody, or maybe a mini proteins or diabodies or mini bodies and things like that. And these are because most of the users are using phage display, different kinds of panning, or small molecules, peptides, fabs, nanobodies, in order to find these building blocks that they are discovering against multiple targets. And once they discover this, target specific small blocks like Lego pieces, they are bringing in 2 different or 3 different kind of blocks, and then they are trying to conjugate it with a FC or FC-like structure. So in order to address this changing landscape, I will showcase some of our biosensors, which are very unique, and also instruments that how people are using and how we have also learn to use in order to facilitate this multispecific antibody discovery. Now, for our Gator family, we have currently in the market 5 different instruments. For today's talk, I'm going to only talk about the Gator Pivot and Gator Pro. These are our 2 high throughput systems. Now let's meet the Gator Pivot, which is our 16-channel automation, ready to install automation build system, and it has the unique advantage of having 2 384 well plate at a time in the instrument. Then you can see that you have full access to these 2 plates, thereby increasing your throughput even without adding any additional automation to it. Along with that, what it has is it's a very stable system, giving you very solid baseline because it's very tight in terms of temperature control. And you can also control the temperature sensitivity of the experiments that you carry out on this machine, starting from 15 degrees centigrade all the way to 14 degrees centigrade. And another thing is that all the BLI instrument, of course, has an advantage that it does not require any fluidics inside. So, of course, there is no regular maintenance and all those things that you have to worry about. But on top of that, well, how we have built Gator Pivot is that it has an automatic picker cleaning option. Thereby, you can constantly run the instrument without having to worry about any kind of maintenance and requirements and slow down of your performances in terms of assay setups and things like that. Another thing that the Gator Pivot we have designed for it is a patented evaporation prevention shield that only will open when the well is being used to do the experiments, because when you are having this large amount of samples, the experiments tend to take longer time, so you would see evaporation. So this is very useful when you have automation set up and you are running experiments for 5 6 7 hours or even 24 hours at a time. Now to show how the sensitivity of the Gator Pivot experiment, here's a small one example where we tested an antigen antibody pair at 2 different temperatures, 20 degrees and 40 degrees. And you can see just by looking at the sensogram that the off rate and the binding constant, that how different they look. And it of course showcases that even though it is a 16-channel instrument, but we haven't compromised in terms of its sensitivity to give you refined data in terms of kinetics information or anything. Now the next high throughput system we have is even higher throughput compared to the Gator Pivot, which is a Gator Pro system, and it is a 32 dedicated channel and no multiplexing spectrometer. So each channel has a dedicated spectrometer, so thereby there is no quality compromise on any data output. And on top of that, the Gator Pro system can have 384 well plate at the same time, and without any automation you can imagine the increase in throughput, how it gives you the walkaway time. It also offers high flexibility, so you don't always need to run a 32 experiments dedicatedly, but you can run anywhere from 1 to 32 experiments simultaneously. And because it has these 4 plates that you can run samples simultaneously, this is the only automated platform currently in the market that you can run a quantitation assay of more than 1100 samples in less than 2 hours without any automation. And if we compare, our 8-channel system is one of our basic system with Gator Pro, and if you look into the tiny detail of the data acquisition, the KD that we can calculate the binding constant here, nothing changes just because the system becomes high throughput. So it indicates that even though the systems are high throughput, but from Gator we have not compromised in terms of the data quality and also the basic stability of the instrument or anything. So there's no compromise getting higher throughput data, but very good data for both the Gator Pro and the Gator Pivot systems. Now, another important component, as I mentioned earlier, is the biosensors. So along with having very high throughput, which are very high sensitive instruments, we also have designed, especially because we have the proprietary chemistry and special optical coating, we have been able to design biosensors that has set us apart at Gator Bio compared to our competitor. So we do have the, traditional, all the biosensors like protein A, protein G, then anti-human FCs and things like that. But along with that, when we, saw our users using nanobodies, SCFVs, small fragments, based peptide, we realized the importance of having high sensitive biosensors so that our users can carry out these experiments directly in order to think about, like, alternative ways to use the machine. So for them we designed anti-VHH biosensors, where you can capture the nanobody directly. We also have, we thought about, like, what are the new tags, and we are compared to the existing tags. So we realized that twin strip tag and anti strip tag 2, these are very popular right now in the markets. We have also directly, you can use this strip tag 10 xt and anti strip tag 2 in order to utilize this new kind of tags. Along with that, we also have understood the importance of peptide and small molecule for this multispecific drug discovery. So we have enhanced streptavidin surfaces like SAXT and SMAP that are designed specifically in order to detect this very small molecule signals with very low to almost no non-specific binding, and that is only possible because of our proprietary chemistry. Now, just to give you a few examples of this, here is an example where we have, showcasing that how can you do quantitation of high throughput in a high throughput fashion of nanobodies. So here we use our streptactin XT probes, load a protein that has twin strep tag. Now the nanobodies are in supernatant, and they are only specifically being bound to the surface here by giving you the quantitation of the nanobodies. Because of our proprietary chemistry, we have minimized the non-specific binding interaction. So these kind of matrices that we get our nanobodies or SCFVs after the phage display and things, we are in Gator, we are able to easily have low to no non-specific binding signals because of our optical coating and also our surface chemistry. Now, another example here is we have screened like a SCFV, but it's also in media, and what we are trying to do is we are trying to do off-rate ranking, and this is done in Gator Pro, and as you can see, it's a 96 well plate. When it does bind to its antigen, you can see the binding pattern, association and dissociation, but then some of them which does not show any binding, you can see that there barely is any signal. So even though these SCFVs are in some kind of media here, but the media, we are able to completely have almost no to very low non-specific binding signals from this media, giving just the specific interaction between the antigen and the SCFV. And this kind of experiments helps in order to understand our users at a very early stage, how to do the lead selection, narrow down on their lead selections and things like that. Now in the next example, what I show here is again nanobodies, and these are loaded onto our anti-VHH probes, And we have the antigen, and we are doing a detailed kinetic binding interaction in order to understand the binding constant and also the dissociation constant in order to rate them or have a lead optimization. So not only that we can get a single point information, but we can also get multi-point information in order to do the lead optimization, the selection and maturation of all these small building blocks that leads up to the multi-specific antibody discovery. And here is an example of our SMAP probe, which I chose here to show that you can see how sensitive the SMAP probes are. Here we have an example of a small molecule which is 330 Dalton, and this one is 332 Dalton. In the traditional BLI, we hardly see any binding in both the cases, but here, after doing the background subtraction, we do see specific binding interaction. So, of course, the proprietary chemistry and how we have built this biosensor allows it to be so sensitive in terms of doing small molecule drug discovery. Now, when we talk about small molecule, another thing that comes another very popular targets are membrane proteins. So recently what we did is we did a collaboration where we tested different membrane proteins expressed in different formats in detergent, nanodisk, as well as VLPs, and we got biosimilars against this GPRC5D, and you can see that how we obtained the association and dissociation of the biosimilar at different formats. So these kind of information helps you in order to set up when you are doing membrane protein screening as to what format would you need in order to get the best data. And also this showcases that the versatility of the Gator biosensor and the instrument, that not only are we sensitive in terms of the previous slide, which is the small molecule, but we can at ease do characterization with this large, very sought after membrane protein in the discovery process. Now, if I summarize what I talked today, is showcasing that I try to showcase the advantages that Gator Bio provides in terms of compatibility with different kinds of complex biological matrix. There, because of proprietary chemistry, we have we are able to reduce the non-specific binding to almost no non-specific binding, thereby allowing you to only have specific interactions. Then with our instruments, of course, we have facilitated high throughput kinetic analysis. And another thing that I forgot to mention is all the biosensors. It's not one-time use. Most of them you can regenerate and reuse over and over again. So it becomes very cost effective. And at the same time, I showed only few examples, but we have almost 35 plus biosensors that gives our users a versatile biosensor toolbox for them to play with in order to optimize their assay and conditions, that whatever works best for them, they would be able to select from those. And the last but not the least is, of course, easy to use software, because if you're a user, you understand how it's very easy to set up our assays as well as to analyze the data with few clicks, thereby giving you this high throughput data information in a very short amount of time. Now, thank you again, all of you, for listening to our webinar today, and please stay in touch. And if you have any questions, we will of course answer at the end of the session, but also you can email us at info@gatorbio.com. Thank you very much.
Well, thank you, Gator Bio, our partner in hosting this webinar. And thank you to the Informa Connect team, as well as the Antibody Society, for organizing this event and for having me. I am honored to speak on behalf of our company, AvantGen, on what we are doing in advancing antibody discovery and development, especially in the immunotherapy space. Today, I will speak about an area that is a nice intersect across both cancer and autoimmune disease treatment, and the intricate means to develop such novel biologics are shared. I truly hope you will gain some research ideas or insights along the way. A little bit about us. AvantGen is a CRO based in San Diego, California, focused on protein and antibody discovery and engineering for our clients. Our clinically validated and next generation libraries have identified unique and superior antibodies that are both distinct and more effective than those obtained through immunization or phage display, and has led to some interesting immune cell engagers that can apply to both cancer and autoimmune disease. So diving right in, immunocell therapy, especially targeting cancer, has come a long way. And yet, in less than a decade, we now have 7 approved CAR T cell therapies that provide a relatively durable response. Now, having said that, all of them are autologous, which is on a patient-by-patient basis, and does not help with the manufacturing ease and associated costs. Cytokine release syndrome is also a concern, and hopefully current gene editing technologies can be a solution to a truly off-the-shelf therapy. What has quickly followed are bispecific T-cell engagers, the protein version of CAR T, with more than 10 approved cancer drugs to date. In general, they are relatively easier to produce, off the shelf, but the half-life requires regular infusions for patients. The next modality has been CAR-NK, which harnesses several advantages of NK cells. The lack of HLA matching, having both innate and adaptive immunity roles. They're off the shelf and generally safer. Relative efficacy and durability is still early days. And naturally, multiple NK cell engagers in the form of bispecific and even trispecific antibodies are in clinical trial, targeting both liquid and solid tumors, as they seemingly have the best features of the previous 3 modalities. One such company spearheading NK cell engagers is Affimed, who earlier last year announced positive phase 2 results with their CD16A EGFR bispecific antibody in combination with Roche's PD-L1 inhibitor for non-small cell lung cancer patients. What is remarkable is that some of these patients with good outcomes were previously treated with checkpoint inhibitors and had developed resistance. And just last month, they presented results showing a higher dose correlated with higher activity response while maintaining safety. I would like to note that I do not have any shares or stake in Affimed. Developing antibodies targeting CD16A to engage NK cells is certainly compelling. Some key points here on CD16A. It is expressed on NK cells and has 2 isoforms. The closest family member is CD16B, which is expressed on granulocytes like neutrophils, and also has 2 isoforms called NA1 and NA2. This, among others, is important to consider for the desired or target antibody profile, which is we want high affinity for both CD16A alleles. However, it should not bind to CD16B, NA1, or NA2 as much as possible. This is an important consideration for 2 reasons. One, neutrophils are the most common white blood cell and would act as a sink and absorb most of the circulating drug if it was to bind to CD16B. And secondly, CD16B on neutrophils has been reported to negatively regulate ADCC, one of CD16A's function. There are also functional and cross-reactivity requirements to consider as well. Now, when we align the amino acid sequences of the relevant target and non-target proteins, here lies the crux of the problem. The extracellular domains of CD16A and B have a 98.5% sequence identity. It's really only at 3 amino acid residues shown here in green are where they are the same in both alleles in CD16A and different in both CD16B isoforms. Now, when we consider the appropriate methods for antibody discovery, we would argue that in this situation, any animal immunization-based discovery efforts would fail from the start, or at best, might succeed as an inefficient and resource-intensive method. The first challenge of immunizing, let's say, a transgenic mouse, would be to obtain a robust immune response, as FCRs are also present across species. If we are lucky enough to have an FCR knockout mouse on hand, or we break immunological tolerance and obtain a strong immune response, the second more significant challenge would be to determine from serotiter the presence of CD16A specific antibodies, as the majority of antibodies raised will likely not be of interest and will mask any interpretable signal. Also, if the regions common to CD16B are just slightly more immunoreactive, which we wouldn't know at this stage, it would make an already needle in the haystack type of intensive screening campaign even worse. Personally, I would not send our scientists down that hole. At AvantGen, our discovery platforms can efficiently tackle a wide range of targets, including difficult ones like CD16A. Our germliner library collection are very large yeast display libraries containing fully human antibodies that are displayed on the cell surface using a novel and proprietary technology independent from other companies or labs using AGA1 or AGA2. For each campaign, we introduce more than 100 billion yeast clones, and we have it in 3 antibody formats. Our AvantEpe, which is our single-chain FV library, our AvantFoil or VHH heavy chain only library, and our AvantSaber Fab formatted library, which is what we used for this NK cell engager program. Our antibodies obtained with our discovery platform will meet the desired target antibody profile in expression, affinity, specificity, stability, immunogenicity, and other developability requirements for 3 critical and interdependent reasons: design, display, and screen. First is design. We deep sequenced over 500 individuals to understand the antibody immune repertoire and formed AvantGen's human antibody database. With this data, we gained insight on the general frequency of all amino acids at each CDR position for each antibody framework. The figure shows the distribution of a particular variable region heavy chain subfamily, where the germline amino acid is shown here, underlined in red, and variants observed and its frequency are represented on the y-axis. For example, for this particular variable region subfamily, CDR2 position 55 has a glycine as a germline, with a 40% observed frequency of having an arginine instead. Positions 56 through 58 following that position are 2 isoleucines followed by a proline with little to no variation present, or less than 5%. This is likely due to evolutionary and functional reasons. If we were to completely randomize or even partially randomize these positions, this could lead to problematic or immunogenic antibody sequences. And it would also dilute the overall functionality of the library. So we don't. Another aspect of our design is that we use 100% germline framework regions, meaning no mutations in the regions between the CDRs, even if it was observed in our data set. Now, framework mutations can naturally occur, but and some of them could be beneficial, but that mutation could also be immunogenic in a general population, especially if it were to be a drug. And so just to be safe, we keep it 100% germline. And lastly, on the design, we also removed most potential problematic sequence liability motifs, such as deamidation, oxidation, free cysteines, etc. We then constructed synthetic libraries where each CDR amino acid position would have these general population weight distributions and diversity. Because of the combinatorial diversity within each CDR, as well as the combination across 3 CDRs per chain, and also the combinatorial pairing between heavy and light chains, well, there's a lot of combinations happening. But importantly, with this rational design and method of construction, we find a practical way of introducing sufficient natural CDR diversity and quote unquote rearrangement to be able to obtain high affinity and developable antibodies without the need for further affinity maturation. Before I elaborate on the second and third critical steps of our platform, I would like to quickly share an example of a panel of antibodies from a single discovery campaign where all the melting temperatures are greater than 80 Celsius and are more thermostable than trastuzumab, emphasizing our germline and high developability design. Here is the same panel of antibodies compared to a fully human FDA-approved antibody, where our antibodies are scoring even higher in humanness by sequence. And this is also based off both the germline framework sequences as well as the natural CDR sequences that we designed. Okay, our second critical factor, display. A library is only as good as how efficiently each and every antibody can be displayed on the cell surface. As a eukaryotic organism, yeast can properly fold and express proteins such as antibodies with similar post-translational modifications as mammals, and display all antibody framework families evenly. Bacteriophage, on the other hand, has been shown to have biased and limited display across different frameworks, and translational risk when eventually producing hits in HEC293 or CHO cells. Now, this might upset those using phage for therapeutic discovery, including myself, admittedly, 6 years ago. But by beginning your screen in an inferior system, not only reduces the availability of good antibody clones to begin with, but the top hits will also have inherent expression and developability risk. And the third critical factor for making this all work, screen. Utilizing successive rounds of magnetic sorting, MAX, as well as fluorescence-based sorting, FACS, we can rapidly narrow down that pool of 100 billion clones to a panel of just 20 or so functional antibodies in a matter of weeks or months. The quantitative sensitivity that comes from FACS allows us to enrich and identify the desirable clones rapidly and efficiently. So we did just this with CD16A. We performed multiple enrichment sorts for the 176V and F alleles, as well as a subtractive sort to remove CD16B binders. Our display system uses a novel engineered peptide that tethers the antibody fragment to the yeast cell surface. Upon a simple switch in selected media, we can induce the secretion of Fabs to use for ELISA and flow cytometry analysis without any need for additional molecular cloning or the addition of reducing agents, which simplifies the screening. A panel of antibodies were indeed confirmed to have the desired specificity. And as shown here, the kinetic binding affinities of our top clones had very high specificity, as well as higher affinity to CD16A compared to a reference CD16A antibody that's currently in the clinic. Our CD16A antibodies have been extensively characterized in vitro, and most data are in the public domain. So in the next few slides, I will just highlight some of the more interesting findings. Remember the sequence alignment that was shown earlier? Well, in determining the epitope for our CD16A antibodies, it's rather straightforward, as there are only several amino acids to look at. We produced single amino acid substitution variants for CD16A at two positions highlighted in green, the G147D, as well as the tyrosine 158H. And we reverted it to what it is in CD16B at those positions. A CD16 pan-AB control clone was able to bind to wild type as well as these two variants equally by ELISA, confirming these proteins were intact. And here, clones A21 and A23 lose binding to the Y158H variant by more than 100,000-fold, meaning that this single tyrosine at position 158 is the single and critical residue that confers these CD16A antibodies ultra specificity. In our very large yeast display libraries, we have more than just ultra-specific CD16A antibodies, as we have observed this phenomenon in multiple discovery campaigns. Our top clones, in addition to reference antibody, were formatted into a CD19 bispecific NK cell engager, or BiKE. In a functional ADCC-dependent tumor cell killing assay using primary NK cells co-incubated with antibodies and a CD19-expressing NOM6 ALL cell line, we observed low picomolar potencies and slightly improved activity compared to the reference. In a second functional assay, these BiKEs were found to be potent in activating JERCAT cells expressing CD16A, the 176V allele, only in the presence of CD19 expressing Raji cells. Clones A21 and A23 were more potent, in fact, than the same anti-CD19 as a monoclonal antibody, but with an ADCC-enhanced engineered FC that is also currently used in clinical trials. Furthermore, A21 and A23 potency is retained even in the presence of physiological simulating conditions of 10 mg per mil IgG, while both FC enhanced and the reference lose almost all of their activity. So this unique property is a key difference of our NK cell engagers in that they do not compete with IgG binding. Okay, so we recently completed an in vivo study using a humanized mouse lymphoma leukemia model, and we're happy to share our findings here. So just briefly, the model we used is a NOD NCG immunodeficient mouse strain, and they are injected with or implanted with human CD34 positive hematopoietic stem cells. They are then boosted with IL-15 to differentiate the HSCs to human NK cells, and roughly 1 week later, the tumor engraftment with the ALL, we engraft with the tumor cell line, an ALL cell line that contains a luciferase reporter gene that can be used to detect the presence of the cancer cells in the mice. So here we see the first time point of the control group. and we're dosing it at this is the higher dose concentration at 15 mg per kilogram with the isotype control. And this is day 27. You'll see here that all the mice, we can see the ALL tumors being circulated, but also present in mainly the spleen and the liver, as well as also for especially mouse 2 and 6 in near the leg. And we can also see remnants of it at the site of injection in the tail. In comparison, at the same time point, with our treatment group, you can see most of them have very reduced or faintly visible tumors present. This is the control group 1 week later, and as you can see, it's quite obvious that the tumors have expanded quite rapidly just within a week, and this is expected as this is a very aggressive ALL humanized mouse model, and due to ethical protocols. This had to be the last time point for the animals, and was our final. They had to be taken down at this point. And then comparing to the same treatment group as shown earlier at day 34, here where we can see, we were expecting almost complete tumor regression, and we see that more or less in animals 2 and 4. However, it's noticeable in some of these mice, particularly in mouse 1 and 3, the tumors continue to grow. We performed immunocytometry work upon takedown and noticed that the number of NK cells present in both the control and treated groups in both the peripheral blood and the spleen tissue were significantly lower than what it is normally in humans, as much as 10-fold lower, which is somewhat disappointing. The silver lining here is that even with an unoptimized humanized mouse model with lower than expected human NK cells, coupled with, as I mentioned, a very aggressive B-cell tumor model, we were still able to see significant bioactivity in our NK cell engager molecule. And since this was our initial in vivo study, I think we have a good path towards a subsequent study. In summary, AvantGen's novel yeast display libraries can identify unique and superior antibodies against a wide range of targets, including challenging ones. Our NK cell engagers have unique properties and activities, and at least in the studies performed, outperformed a first-in-class CD16A antibody in the context of cancer. We have also thought about how this translates to autoimmune disease, as we all know that there are ongoing clinical trials utilizing NK cells. In one case is to deplete overactive B cells that express autoantibodies and inflamed tissues in lupus and other autoimmune disease patients. And so our NK cell engagers should naturally extend here as well. And rather than having only weeks to months to avoid terminal cancer stage, autoimmune-related lupus patients in particular are looking to manage disease with medicine that have better overall safety profiles, which I think might be a good fit in this area. So that is the conclusion of my talk. Thank you so much for your time and consideration and your attention. I also want to thank our incredibly hardworking team at AvantGen and all of our partners. Thank you very much.
Thank you both. We're now going to begin answering the questions submitted during today's presentation. Our first question looks like this one's for Jordon. In your mouse study, you mentioned the human NK cell population was lower than expected. How about other cell types?
Right, Chris. So, in addition to quantifying the number of human NK cells, we also looked at human lymphocytes and T cells in particular, as well as both CD4 and CD8 subpopulations. And what I can tell you, at least from our in vivo partner, as I'm not the expert here on this, that the T cell populations were quite abundant, and it's very likely that their growth dominated the NK cell population in particular. Also, when choosing this, looking at it now, when we chose this particular humanized mouse model, it seemed it would be good to have the presence of all these cell types just to see how it plays out, especially since CD16A is also present on macrophages, which could also have an interesting role here. But in hindsight, I think a human NK cell only model would have been a more focused initial study to obtain more clear-cut proof of concept data.
Thank you, Jordon. And going through a few more questions are coming in here. Looks like the next one is for Indrani. What advantages does BLI offer over traditional ELISA or SPR in screening multi-specific antibodies?
So, let me first compare with ELISA and BLI. So for ELISA, as we all know that it is a endpoint assay, whereas BLI is real-time measurements. And with BLI, you get the K-off rate constants. You also can determine concentrations in the same experiments. And if we set up a little bit diverse experiments, we can get more information in terms of epitope, cross-reactivity, etc. So if I just compare BLI with ELISA, then you can see that as well as getting real-time binding data, we can also have different other information from same assay. Now, if I compare BLI with SPR, both are label-free real-time binding experiments. But the one thing that differentiates between BLI and SPR is the presence of the fluidics in the SPR instrumentation. So when you are doing screening, you normally are dealing with a lot of crude sample, lysates, cell lysates, and things like that. And these tend to clog very often the SPR fluidic system, thereby not allowing that much of throughput capabilities, whereas that is totally not present in BLI and allowing very easy throughput and high quantity data information.
Thank you. And the next question we have is, this area of NK cell engagers is so fascinating and new. Where do you think this class of immunotherapies will be in the next 5 years? Jordon, I believe that one's for you.
Oh, wow. Okay. 5 years sounds like a lot, right? It's difficult, I feel like, these days to even predict what's going to happen in a year. But in terms of the typical R&D cycle, the average time from discovery to approval is still around 10 years, I would say, and probably shorter with newer regulations and technology in the future. So in a 5-year period, we should have fairly solid data on the efficacy of these first-in-class therapies for cancer in both liquid and solid tumors, but also in autoimmune diseases as well. Importantly, though, I could see this class of drugs follow similarly to the history of ADCs and CAR T, where likely second generation or even third generation class of immune cell engagers will also enter clinical trials by then, and will likely have even more potential. So very promising.
Great, thank you. And the next question we have is for Indrani. What types of multi-specific antibodies can be effectively screened using Gator Bio's platform?
So, multi-specific antibodies have, as I was discussing during my presentation, different kinds of formats. And if you imagine, as I mentioned, that it is more like Lego blocks. So you can have nanobodies, peptides, small molecules, Fabs, SCFVs, different kinds of formats. So the advantage of the Gator Bio BLI platform is we have high sensitive different kinds of biosensors that can be used to capture and test specific binding of these building blocks for multi-specific antibodies. So at the very end of a project, how you can use BLI. Similarly, when you have now chosen your lead and moved forward in order to, you have already determined your top 10 contenders, then you can do a thorough analysis. That also you can do a detailed kinetic analysis. Using special mention is our Gator pivot system that is very sensitive to temperatures, where you have informations that how it binds at different temperature and how different kind of temperature affects it. So throughout, I think I had one of my slides during the presentation that I mentioned that from the very beginning or the conception of your multispecific antibody to the end, almost clinical samples, all of it can be used in different assay formats using the BLI principle with Gator Bio to evaluate its efficacy.
I just want
Wonderful.
to add to that
Mhm.
because in our presentation, when we screen our panel of bispecific antibodies, we use the Gator Bio instrument, and it works very well and efficiently. I think the one thing, the data that we didn't show just because of time constraints was that it also worked very well when we were using it for epitope binning. So that was makes it very clear to see, what shared epitopes they have or have not. So thought that was also very helpful.
Wonderful. Thank you both for your response. And the next question, this one's for you, Jordan. It says, it's incredible to see your antibodies that are specific to a single amino acid difference. I wonder if that is necessary for most therapeutic applications.
Ooh, that's a really good question. Well, I would say in general, it's probably better to overperform than underperform, right? Uh, you'd want to at least demonstrate, right, that the platform has a wider range of solutions, especially for a multitude of challenges. And in terms of drug discovery, having higher specificity applies to a number of applications, right? Such as personal medicine, recognizing disease-specific mutations over wild type, mutant isoforms, splice variants, even like MHC peptide-specific combinations, right? So the list goes on to have more higher specific antibodies. So having these types of antibodies, it's one way to address this, right? Without having to engineer the antibody. But of course, there are other ways, right, to engineer the antibody to be more specific, like making them conditionally active, right, with some engineering work. But I think ultimately, right, having a combination of these unique properties will really bring about the superior and next gen medicine.
Great, thank you. And Indrani, this next question is for you. How does BLI handle simultaneous binding of multiple targets in multi-specific antibodies?
That's a good question. So for BLI, we can only have one interaction characterized at a time, but certainly you can set up different assay formats to evaluate your multi-specific antibody. So if you have, say, for an example, a bispecific antibody, you can capture simultaneously using two different sensors, your two different target antigens, and then show that how the particular bispecific antibody binds to one antigen versus another, determine their K on, K off, and KD binding constant. And so this information you can seek. Certainly, it's not a multiplexing format where you would simultaneously gather the multiple antigen binding. That is definitely it's not available. But you use the BLI through its strength to understand the kinetics with different binding partners, to understand its epitope specificity against the targets and things like that.
Great. Thank you, Indrani. And it looks like this next question is for you as well. In what stages of antibody discovery or development is Gator Bio's BLI screening most beneficial?
Screening, if you're talking about, of course, when you have a lot of samples. So right now, a lot of them are using the AI de novo models, as well as phage displays, antibody engineering. So that is when you have like almost thousands of samples. So that is when the Gator Bio's Pivot and Pro platforms come in handy. As I mentioned during my presentation, they can also be equipped with automation, and you just have very automated experimental setup to do your initial screening. I hope it answers the question.
Yes, thank you. And so at this point, I'd like to say thank you to our speakers, Indrani Chakraborty and Jordon Wang, and thank you everyone for attending today's webinar. On behalf of Gator Bio, AvantGen, and our presenters, thank you for joining us today and have a great rest of your day.
That's all the time. Thank you, Jordon and Indrani, for a great session. If anyone submitted a question that wasn't addressed, keep in mind that the speakers will reach out to you directly. This session was recorded. You'll receive a notification in 24 hours when the on-demand session is available for viewing. And before you log off, please take a moment to complete the feedback form so we can continue to improve your Digital Week experience. On behalf of Informa Connect Live,